Agriculture in the classroom seeks to improve student knowledge of agriculture by applying authentic, agricultural-based content as the context to teach core curriculum concepts in science, social studies, language arts, and nutrition. In an effort to further deepen the partnership from the grassroots, Cabinet Office directed Executive Office of the President on Tuesday the 17th of September 2024 that the Ministry of Environment, Climate Change, and Forestry to establish a more robust framework for community-based forest associations to further empower local communities in forest conservation efforts. National Landscape and Ecosystem Restoration Strategy 2023 to 2032, which aims to plant 15 billion trees. Cabinet acknowledged with appreciation the growing partnership between the public and private sectors in advancing the tree planting campaign. Welcome to Suya e-learning for food security, climate change, school feeding program, nutrition education for all. When you have bad governance and poor accountability from your local government leaders, then you are doomed to live under control of cartels and fools. We hope you subscribe to our channel YouTube at Suya Mombasa and share this links with your friends or family. Saturation refers to a soil's water content when practically all pore spaces are filled with water. This is a temporary state for well-drained soils, as the excess water quickly drains out of the larger pores under the influence of gravity, to be replaced by air. What happens when soil is saturated with water? Saturated soil is waterlogged with all its air squeezed out. If soil stays saturated too long, it can promote devastating diseases, leading to the death of trees, shrubs, and other plants. Healthy soil has little pockets of air between the molecules of sand, clay, silt, gravel, and rock. Water moves through a soil in the large pores and drains away due to the force of gravity. This water is called gravitational water. Gravitational water represents the difference in soil moisture content between the saturation condition and field capacity. What do you mean by gravity water? The amount of water in the soil that percolates downward under the influence of gravity and reaches the water table is called gravitational water. whereas the amount of water that is retained in minute interstitial spaces in the form of thin films surrounding the soil particles is known as capillary water. Capillary water is held in pores that are small enough to hold water against gravity, but not so tightly that roots cannot absorb it. This water occurs as a film around soil particles and in the pores between them and is the main source of plant moisture. As this water is withdrawn, the larger pores drain first. Hygroscopic means the tendency of absorbing moisture from the air. Some water in the soil forms an extremely thin, tightly held film around the soil particles. It is called hygroscopic water. In the soil, a small portion of soil water is chemically bound with soil materials. It is called combined water. Composting is a controlled, aerobic, oxygen-required process that converts organic materials into a nutrient-rich, biologically stable soil amendment or mulch through natural decomposition. The end product is compost. Microorganisms feed on the materials added to the compost pile during the composting process. They use carbon and nitrogen to grow and reproduce, water to digest materials, and oxygen to breathe. You can compost at home using food scraps from your kitchen and dry leaves and woody material from your yard. What is the golden ratio for composting? The optimal ratio for a compost should be roughly 70% carbon, leaves, pine straw, wood chips, nut shells, and 30% nitrogen, vegetable scraps, garden weeds, coffee grounds, manures, hay. Here are the carbon to nitrogen ratios for some typical materials. Poultry manure, from 3:1 to 15:1. Cow manure, 20:1. Horse manure, from 20:1 to 50:1. Food waste, about 15:1. Fresh grass clippings, 15:1. Sun-dried grass clippings, 20:1. Oak leaves, 40:1 to 80:1. Straw, 50:1 to 150:1. Sawdust, 200:1 to 750:1. Newsprint, 400:1 to 850:1. Corrugated cardboard, about 560:1.
Mealybug is an important insect pest of cassava in Thailand. Adult mealybugs can lay 100 to 500 eggs in egg sacs, which develop into the first nymphal stage within 8 days. The nymphs molt 3 times, so the insect has a life cycle of 21 days. The mealybugs are transferred from plant to plant by ants or wind. The insects dwell under the leaves and suck plant sap. Mealybugs spread in dry and warm conditions. They are able to damage cassava fields in a short period of time. As an alternative to chemical insecticides, Entomopathogenic fungi, including Beauveria bassania, can be used as a natural means of insect control by using topical application of fungal spores. Beauveria spores land on the mealybugs. With high humidity, spores germinate and penetrate the insect cuticle. The early infection cells, called hyphal bodies or blastospores, are formed inside. Inside the insect body, the innate immune system is ready to defend against the invading pathogens. Insect hemocytes would be a major line of defense. On the fungal side, secondary metabolites play crucial roles in insect pathogenesis, such as avoiding the insect immune response, or as toxins for killing insects. Our previous phylogeny identified 2 polypeptide synthase genes highly conserved for entomopathogenic fungi, including PKS3 and PKS2b. We are also studying the roles of a non-ribosomal peptide synthase gene, FER-S, in insect pathogenesis. PKS3 consists of ketosynthase, acetyltransferase, and acyl carrier protein, the 3 principal domains. PKS2b is similar to PKS3 except it does not contain the anoyl reductase domain. The non-ribosomal peptide synthetase, FER-S, is arranged in a modular structure. Each module consists of adenylation, thiolation, and condensation domains. We studied the function of the 3 genes by disrupting each gene with a selectable marker. When the selectable marker interrupts the DNA strand, the translated protein becomes truncated and non-functional. You can see that the tie boxer is now tied up, the soldier's gun is broken, and the drill is broken. Fungal PKS3 presumably acts to cope with the insect immune response. In the wild type, the fungus can overcome the insect defense and the infected insect dies. The PKS3 null mutant, in which this polypeptide synthase is not working, cannot cope with the insect immune response, so the insect remains healthy. We determined the insect virulence between Beauveria wild type and the PKS3 null mutant using intrahomocoelic injection of fungal spores. The results showed that the wild-type injection resulted in 100% death, whereas injection with the mutant spores caused only 60% of the insects to die. The amoeba, used as a model, has analogous properties to the insect hemocytes. The amoeba often phagocytosed only 1 to 2 wild-type blastospores. The blastospores survive within the amoeboid cell and grow out, which leads to the amoeboid's death. By contrast, 3 to 5 of the PKS3 null mutant blastospores could be phagocytosed, and most were degraded by the lysing enzymes. Here, we have demonstrated that the mutant is unable to suppress the insect immune response, in this case, phagocytosis by hemocytes. PKS2b might work together with PKS3 in defending against the insect immunity, or it could be a toxin that can subdue and kill the insect. Similarly to the PKS3 study, the PKS2b null mutant cannot help the fungus to overcome the insect defense or does not produce the toxic molecule, so the inoculated insect survives. We tested the insect virulence in a similar way to that conducted for the PKS3 study. Only 40 to 80% of insects injected with the PKS2b null mutant died. Again, 100% of wild-type injected insects died. Most likely, PKS3 and PKS2b genes from Beauveria are involved in suppression of the immune response system of insects, while FER's gene may associate with fungal germination.
In this video, we are diving into the incredible world of Trichoderma, a beneficial fungus that can do wonders for your garden. We are going to explore 5 unique ways to use this organic biofungus for your plants. Stay tuned because point number 2 is a game changer for starting seeds, while point number 5 unveils the correct and most effective way, and my personal favorite one, to use Trichoderma in our garden that you won't find anywhere else on the internet. Let's start with what is Trichoderma. It is a naturally occurring soil fungus. It can also be cultivated in lab and usually comes in white powder-like structures. Although Trichoderma belongs to a broad family of fungus that comprises more than 100 species, but only few of them, like only 5 species, are reported to be useful as biocontrol in gardening. And among them, Trichoderma viride and Trichoderma virginum are the most common to use in agriculture. You can easily get this in local gardening shop or online. I'll put some links to buy it online in the description box. It's always better to check the manufacturing date before buying. The manufacturing date should not be more than 6 months when you are using it, as the population and activities of this useful fungi degrade over the time. Also, you need to store it in a cool place away from direct sunlight. Trichoderma is used for different purposes in agriculture. It interacts with other microorganisms, but mainly with the pathogenic fungi or the bad fungi in the soil. These interactions include the competition for food, nutrients, and space, and as a result, it can suppress the activity of those bad fungi by creating unfavorable conditions for them. Thus, the application of Trichoderma in the soil can provide resistance to different diseases, mainly soil-borne fungal diseases like wilt, root rot, and damping off. Apart from that, the Trichoderma has the ability to degrade complex organic compounds present in the soil. It can convert these organic compounds in the simpler form so that it would be easily absorbed by the plant. In this way, the use of Trichoderma in your garden can improve crop yield and performance. That's why it is widely used as a biofertilizer almost for all crops with or without additional soil amendments. It has been also reported that the Trichoderma has the ability to degrade the chemical contaminants present in the soil. Heavy metal contaminants like nickel, cadmium, arsenic, lead, and zinc can be accumulated by Trichoderma species, and thus it can significantly improve the physical and chemical properties of the soil. However, remember that if you are applying Trichoderma in the soil, you cannot use any chemical fungicide directly into the soil, as it will kill those useful fungi population. Now let's see what are the 5 ways to use this Trichoderma in your garden. Number 1, to use it while preparing your garden soil or potting mix. As the Trichoderma fungi feed on organic carbon present in the soil, it is best to mix it with organic manure or compost, which are very rich in carbon. Mix 100 g of this powder with 10 kg compost. It could be leaf compost, cow dung compost, or vermicompost. Then add the compost in the soil mix or potting mix. After adding it, water the soil immediately, as the moisture is the essential factor for the growth and survival of this useful fungi. Number 2 is to use for seed treatment. Before sowing the seed, mix 1 gram of Trichoderma with 50 to 100 gram of seeds. Note that such seeds should not be chemically treated. In some seeds, you can find colorful chemical coating. In such cases, mixing Trichoderma won't be effective. Ensure that the seeds are untreated and organic. You can mix water with it and soak the seeds overnight before sowing. It will increase the germination rate as well as prevent the seedlings from damping off or root rot. Number 3 is to use it in the plant directly by soil drenching. Mix about 5 gram of this Trichoderma powder in 1 liter of water and apply it to the root zone. It will be easily uptaken to all parts of the plant, and also it will improve the existing soil quality and prevent diseases from soil-borne pathogens. You can use it twice in a month to get best results. The 4th way of using Trichoderma is to mix it with compost and water and leave it for a day to activate those Trichoderma fungi, and then apply it as biofertilizer. Take about 1 kg of vermicompost, add 20 gram of Trichoderma powder, mix them well and sprinkle water on it. Cover it with some paper and again add some water so that the paper can hold enough moisture. Remember, moisture is the essential factor for the survival of Trichoderma, while the organic carbon present in the compost will help them to grow. Keep it in a shady, cool place away from sunlight. We'll leave it for a day or 2. Actually, the fungi which came in the packet remain dormant, and in this way we can activate them. Then we can directly apply it in the soil as fertilizer. After applying it, water thoroughly. the fungi will be blended into the soil and will establish colony there and multiply over time. However, using all these 4 methods, it will take months for the trichoderma fungi to multiply in the soil. Keep in mind that our objective is to cultivate a substantial population of these fungi. The greater the presence of beneficial fungi in the soil, the more effectively it will counteract the growth of harmful fungi. And that brings our final and 5th method of using trichoderma. And this is also my favorite one. This is very efficient method that allows rapid population within just 4 to 5 days. By utilizing a small quantity of trichoderma, you can generate millions of them using this technique. And this is also more economically efficient. By employing this technique, you can effectively propagate any beneficial fungi, trichoderma, mycorrhiza, or any others. In actual laboratory, scientists use a specialized growth medium known as Japedox, but it is expensive. However, you can easily create a similar growing medium at home using common household ingredients, and you won't find this technique in any gardening videos in the YouTube. Take about 500 grams of potatoes, slice them and boil in sufficient water. After boiling, strain the potatoes. We need this remaining water. Its quantity should be around at least 500 ml. So take the water for boiling accordingly. Let the water cool completely to room temperature. This starch-rich solution serves as the base for growing medium. Add 25 grams of glucose. This food-grade powder is readily available in the market. This glucose will provide all the foods or nutrients for fungal growth. Finally, add 25 grams of trichoderma powder in it. Even this small amount of fungi can multiply exponentially within days. Thoroughly mix the ingredients and cover the container with a breathable cloth to allow proper air or oxygen circulation. This oxygen is vital for fungus growth. Store the mixture in a dark and cool area of a room for 4 to 7 days. Don't wait beyond that. After 4 days, you can see this impressive growth of fungi on the surface. This upper layer predominantly comprises a substantial population of trichoderma fungi, although a few other fungi may also grow as byproduct, but this is not a concern at all, as the trichoderma dominates the population. Thoroughly blend this fungal layer with the solution to ensure most of the trichoderma is mixed in. Next, strain the solution, which now contains millions of beneficial trichoderma fungi. Starting with just a few grams of trichoderma, we have multiplied their population significantly, which is sufficient for your entire garden. Dilute this 500 ml solution with 10 liters of water and apply it to all your plants, mainly in the root zone. A little amount of this is enough for the plant. You can safely use this in the seedlings as well. This active trichoderma will begin their beneficial work in the soil right away. They are known for quickly colonizing and establishing themselves in the soil, where they can help with nutrient uptake, disease suppression, and overall plant health. Keep in mind that introducing this organic biofungus doesn't always assure immunity against fungal issues for your plants. Despite careful measures, changes in the climate or the fungus evolving immunity can still harm your plants. In such cases, chemical fungicide might be necessary, although I advise against their use on vegetables and fruit plants. If you are interested in learning about such 7 common fungal diseases in the plants and their remedies, click the video on the screen. That will be all for today. Thanks for tuning in, and see you next time in another interesting content. And don't forget to like, share and subscribe, and let us know in the comments which trichoderma application you are most excited to try in your garden. Until then, enjoy your gardening journey.
Bioenzyme fertilizers can enhance photosynthesis in plants and increase soil's organic matter. As a result, plants will get more nutrients and their roots can absorb more air. Bioenzymes are key when it comes to building fertile soil. It is a natural fertilizer. It can make a barren land fertile. Fermentation basically produces either acids or alcohols. Acidic fermentation selectively kills acid-intolerant bacteria, and only the ones able to live in acidic conditions thrive. Mostly, fermentation yields lactic acids, acetic acids, and other organic acids. Bioenzyme fertilizers can enhance photosynthesis in plants and increase soil's organic matter. As a result, plants will get more nutrients and their roots can absorb more air. Bioenzymes are key when it comes to building fertile soil. It is a natural fertilizer. It can make a barren land fertile. Fermentation involves the use of microorganisms, like bacteria and yeast, to produce the enzymes. Enzymes energize and catalyze biochemical reaction with high specificity and enhance the reaction rate. Enzymes are formed during growth of microorganisms, tropoplasm, because of oxidative metabolism and aerobic fermentative. This fermentation creates natural chains of proteins, mineral salts, organic acids, alcohol, and enzymes. This enzyme bio-cleaning solution helps to displace unknown, potentially pathogenic, disease-causing bacteria with known, healthy microorganisms, and in this way contribute to better human health. For removing stubborn stains and odors, colored fabrics and floors. Clear blockages in kitchen sinks and drains, use blended pulp sludge of enzyme. As a natural insect repellent, use undiluted, for ants, cockroaches for mopping floors. As fertilizer for plants in garden. Wash cars will look as if they have just been polished. Save money, save space, save water, and save the earth. Improved protocol: a mixture of cellulases from Aspergillus species are used as a pre-treatment to facilitate the release of the cellulose film from the surfaces of the sensors. There will come a time when only those that know how to plant will be eating. Chief Oran Lions. If you like African food systems content, please subscribe and share our channel with your friends and family. Or if you're new here, hit the subscribe button so you make sure to never miss our weekly presentation podcast. Agriculture is tool to create health, wealth, shelter, protect environment, food, nutrition and basic skills development for our local communities.